So today we're going to talk about the second part of lecture 1, which is protein structure and function. So when you talk about molecular biology, usually you talk about the central dogma, the fact that DNA makes RNA, makes proteins, and we organize the syllabus in pretty much that way. However, it's very hard to talk about the DNA without having any knowledge of the proteins, because it's sort of a chicken and egg dilemma, with the DNA being the egg and the proteins, and to some extent RNA being the chicken. Even though DNA is super important and there would be no chicken without the egg, DNA is also incredibly inert. It's non-catalytic, it's protected, and by itself, it doesn't do anything. In fact, you have to have a whole army of proteins to take care of the DNA. And it is the proteins who are responsible for all the processes we're going to cover in the next few weeks, such as DNA replication, transcription, repair, segregation, and packaging. As a result, I wanted to talk about protein structure and very basic protein functions first. Now, in many diagrams you guys have probably encountered in your biology AP class, in Lab 1010, and other classes you have taken in college, and other textbooks, is our oversimplification of the protein structure. The reason for it is to explain the actual underlying process of, in this case, transcription. However, the most important player, the one that actually executes the process, the protein called RNA polymerase, sort of is shoved to the back in order to explain where and how the DNA is located, how it's opened, how the RNA is transcribed from it, and so forth. Now, I hope that one of the important things you'll be able to carry away from this introductory to molecular biology class is that proteins are not color-coded blobs. And it is the structure, which is determined by the amino acid sequence. It is the structure of the protein that will determine how the protein functions. Now, here is the actual structure on the left, just of the catalytic areas, and on the right of the whole shebang of the RNA polymerase. And hopefully you can see how the structure of the protein determines how the protein will function. You can see how, in this area, the multiple helixes of the RNA polymerase are holding the upstream DNA and allowing it to unwind so that the RNA can be synthesized in the active site location. The other thing that I hope that you will carry away from it is that even though the RNA polymerase is portrayed in the previous image as one blob, there's actually multiple proteins that form the RNA polymerase that bind to each other in a very particular way to allow the whole RNA polymerase machine to perform its work, such as the TF2F domain here, TF2H domain right here, and TF2E domain here. And overwhelmingly, proteins proteins in our cells are not single globular boring things, but multi-protein subunit, whether they're homodimers or heterodimers. And the shape and the sequence of amino acid residues inside of the different parts of the RNA polymerase will determine how the different parts work. Which part is responsible for catalysis, which part is responsible for DNA binding, and which part is responsible for moving along the DNA strand as the RNA is being made. And this whole principle of form of a protein following its function, the form being directly related to the protein's function, is something that has been recognized for a long time. And it's found in various obvious ways in other parts of nature, such as in the coloring of this beautiful coral reef fish, that taken out of context would seem garish and strange, but in the context of a coral reef allows the fish to blend in. So its form, its coloration, follows the function that the coloration needs to fulfill, allowing the fish to hide from the shark. And same goes for the architects and designers such as Louis Sullivan, who came up with this very famous quote, which states that that in all true manifestations of life, it is recognizable that form ever follows function, and which is demonstrated here in this picture of stackable tables, which also follows the function that the tables need to perform. They have to be flat on top, so that you can put your martini glass on it, and they have to be stackable, so they have to fit precisely one into the other. So form follows function, people. Now, because proteins are complicated little guys, the way biochemists and biologists and molecular biologists look at proteins is using several levels of complexity. The first level of protein structure is the primary structure of the protein, aka the arrangement of amino acids along the polypeptide chain. Now, the arrangements of the amino acids is super important because the primary structure, how the amino acids are arranged in the polypeptide sequence, will determine all the subsequent structures. You can break the quaternary or tertiary structure of the protein down to the primary structure, and yet the protein will gain it back when the conditions are perfect. So primary structure is just the amino acid sequence. The secondary structure, such as things like alpha helix and beta sheet, are determined by the backbone interactions of the amino acids. The tertiary structure is the folded three-dimensional polypeptide chain, where the location of the different alpha helixes and beta sheet in relation to each other is determined by the side chain interactions of the amino acids. So backbone being this uninterrupted long thing, and the side chains being these guys. And since we have a bunch of amino acids, they have all kinds of different chemical properties, the interactions will be different depending on the environment which they're placed in. And then finally, the quaternary structure is the arrangement of the different subunits inside a multi-part protein, such as the RNA polymerase or other multi-subunit proteins. And that's what you can see here. So you can see that taken by itself, the alpha helix and the beta sheets here are part of the secondary structure. However, their arrangement with each other, how they're angled towards each other, and how they're located, which will actually determine the three-dimensional shape of the protein, is part of the tertiary structure because it's organized by the interactions of the side chains with either backbone or with other side chains. And then finally, this is the final quaternary structure of the RNA polymerase. You can see because several different proteins have been arranged in a very particular way in order for the RNA polymerase to be allowed to be able to do its work. Now, what are the bonds involved in this? So the amino acid sequence is held together by the covalent bonds, peptide bonds, as we've covered last time. The shape of the alpha helixes and the beta sheets is determined by the hydrogen bonding of the backbone. Depending on which amino acids are present in which arrangement, either the alpha helical arrangement or the beta sheet arrangement will be more preferable, more energetically favorable. And you can see the hydrogen bonding here in this picture, in this diagram of an alpha helix. You can see that the carboxyl groups and the amino groups of the backbone of the polypeptide are interacting with each other, forming this very distinct alpha helical shape. Now, the tertiary structure then is determined by the multitude of bonds that the side chains of the amino acid are capable of creating, such as hydrogen bonding, hydrophobic interactions, ionic bonds, interaction with ionic bonds between the basic and acidic amino acids, etc. And then finally, same principles, same multitude of bonds that the amino acids are capable of making will also allow the interaction of the two separate polypeptides in a very particular way, forming the quaternary structure. And it is only the final structure of the protein that will be fully functional. So if you have broken all the bonds, all the connections, all the electrostatic interactions between the backbone and the side chains, and you just have a fully unfolded polypeptide chain, the chain will not be functional. It's lacking the three-dimensional form to fulfill its function. Similarly, just because some alpha helixes have been formed doesn't mean that the functionality of the protein can be achieved, as the alpha helixes and beta sheets should not just exist randomly, but need to be packaged in particular ways in order to form the interacting or the reactive interfaces of a protein. And then depending on whether the protein functions as a monomer, so a single protein, or as it functions as a dimer or polymer of some kind, it's either tertiary structure or the quaternary structure of the protein, it will be fully functional. And all of these steps can be regulated with other small molecules or other proteins. So you can turn a protein on and off even though it's in its full quaternary structure by plugging the binding site, for example.